Graph Triple Attention Network for Disease-Related lncRNA Prediction Ping Xuan, Liyun Zhan, Kui, Tiangang Zhang, Toshiya Nakaguchi, and Weixiong Zhang. Abstract, abnormal expressions of long non-coding RNAs, lncRNAs, are associated with various human diseases. Identifying disease-related lncRNAs can help clarify complex disease pathogeneses. The latest methods for lncRNA disease association prediction rely on diverse data about lncRNAs and diseases. These methods, however, cannot adequately integrate the neighbor topological information of lncRNA and disease nodes. Moreover, more intrinsic features of lncRNA disease node pairs can be explored to better predict their latent associations. We developed a novel method, named GTAN, to predict the association propensities between lncRNAs and diseases. GTAN integrates various information about lncRNAs and diseases, and exploits neighbor topology and attribute representations of a pair of lncRNA disease nodes. We adopted in GTAN a graph neural network architecture with 3 attention mechanisms and multilayer convolutional neural networks. First, a neighbor-level self-attention mechanism is constructed to learn the importance of each neighbor for an interested lncRNA or disease node. Second, manuscript received April 17, 2021, revised November 6, 2021, accepted November 18, 2021. Date of publication November 23, 2021, date of current version June 6, 2022. The work was supported in part by the Degree in Postgraduate Education and Teaching Reform Research Foundation of Heilongjiang University under grant JGXM_YJS_2019032, in part by the Natural Science Foundation of China under grants 61,972,135 and 62,172,143. In part by the Natural Science Foundation of Heilongjiang Province under grant LH 2019-A029, in part by the China Postdoctoral Science Foundation under grants 2019 M650069 and 2020 M670939, in part by the Heilongjiang Postdoctoral Scientific Research Steering Foundation under grant BHLQ 18104, in part by the Fundamental Research Foundation of Universities in Heilongjiang Province for Technology Innovation under grant KJCX 201805, in part by the Innovation Talents Project of Harbin Science and Technology Bureau under grant 2017 ROXJO 94, in part by the Fundamental Research Foundation of Universities in Heilongjiang Province for Youth Innovation Team under grant ROCATED 201805, in part by the Higher Education and Teaching Reform Research Foundation of Heilongjiang University under grant 2019 C29, and in part by the Foundation of Graduate Innovative Research under grant ASCX 2021-199 HLJ. Corresponding author, Tiangang Zhang, Ping Xuan and Liyun Zhan are with the School of Computer Science and Technology, Heilongjiang University, Harbin 150,080, China. Email, zwanping@ju.edu.cn, zinli19@163.com. Kui is with the Department of Computer Science and Information Technology, La Trobe University, Melbourne 3083, Australia. Email, l.kui@latrobe.edu.au. Tiangang Zhang is with the School of Mathematical Science, Heilongjiang University, Harbin 150,080, China. Email, zhang@ju.edu.cn. Toshiya Nakaguchi is with the Center for Frontier Medical Engineering, Chiba University, Chiba 2,638,522, Japan. Email, nakaguchi@faculty.chibu.jp. Weixiong Zhang is with the Department of Computer Science and Engineering, Department of Genetics, School of Medicine, Washington University in St. Louis, Missouri 63130 USA. Email, Weixiong. Zhang@wussel.edu. Digital Object Identifier 10.1109/JBHI.2021.3130110 topology level attention is proposed to enhance contextual dependencies among multiple local topology representations. An attention-enhanced graph neural network framework is then established to learn a topology representation of top-ranked neighbors. GTAN also has attribute-level attention to distinguish various contributions of attributes of the lncRNA-disease pair. Finally, attribute representation is learned by multi-layer CNN to integrate detailed features and representative features of the pair. Extensive experimental results demonstrated that GTAN outperforms state-of-the-art methods. The ablation studies confirmed the important contributions of 3 attention mechanisms. Case studies on 3 cancers further showed GTAN's ability in discovering potential lncRNA candidates related to diseases. Index terms: lncRNA-disease association prediction, graph triple attention network, neighbor-level self-attention mechanism, topology-level attention, attribute-level attention, neighbor topology information. Introduction: long non-coding RNAs, lncRNAs, play an important role in various cellular and biological processes, such as cell differentiation and gene expression. Recent studies show that the abnormal expression of lncRNAs is closely related to the onset and progression of diseases. 1-3. Therefore, predicting the associations of lncRNAs and diseases can help understand disease mechanisms and develop diagnostic and therapeutic strategies. 4-6. Computational methods for lncRNA-disease association prediction have been shown to be effective in disease-related lncRNA candidates screening. 7-9. Some of which have been validated in disease studies. 10-11. The existing computational methods for lncRNA-disease association prediction can be grouped into 3 categories. The first utilizes biological information of lncRNA based on the premise that lncRNAs close to one another in the genome tend to have similar expression patterns and be associated with similar diseases. For instance, genomic loci of lncRNAs are directly used to estimated lncRNA-disease associations. 12-13. Tissue specificities and expression profiles of lncRNAs have also been utilized to infer disease-related lncRNA candidates. 14-17. The methods in the second category use conventional machine learning techniques. For example, a Laplace regularized least square model has been proposed to screen disease-related lncRNAs under the hypothesis that functionally similar lncRNAs are often associated with similar diseases. 18-19. However, these methods only consider lncRNAs and diseases while neglecting 2168 to 2194 copyright 2021 IEEE. Personal use is permitted, but republication/redistribution requires IEEE permission. See https://www.ieee.org/publications/rights/index.html for more information. Authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. 2840 IEEE Journal of Biomedical and Health Informatics, Volume 26, Number 6, June 2022. Relevant information from other sources, such as lncRNA-related RNAs and proteins. Popular machine learning approaches have been adopted to integrate information of lncRNAs, diseases, RNAs and proteins, which include support vector machine, 2021, naive Bayesian, 22-24, non-negative matrix factorization, 25-28, and random walk-in heterogeneous networks, 29-33. These methods, however, are unable to find the underlying complex and nonlinear relationships across the data from different biological hierarchies and different sources. The third category is based on the recent deep learning techniques. Xuan et al. 34. Proposed a lncRNA-disease prediction model based on a dual convolution neural network, CNN, and Wei et al. 35. Fused biometric blocks through deep neural networks to identify the associations between lncRNAs and diseases. ACTN-based heterogeneous network was proposed for drug disease association prediction. 36. Recently, graph convolutional networks, GCN, and variance autoencoders, VE, have been exploited for modeling lncRNA disease association and candidate prediction. 37, 38. Zhang et al. 39. Established a model based on graph convolutional networks, GTAN, and adversarial regularization to predict lncRNA-disease associations. In addition, a computational model based on graph attention network and multilayer perceptron, MLP, was proposed for association prediction. 40. Deep learning models outperformed conventional machine learning models since the former can extract deep relations between lncRNAs and diseases. lncRNA node and its neighbor nodes that have similar functions are often associated with similar diseases. Therefore, the neighbor topology information of a node is an important piece of auxiliary information for association prediction. Unique information of lncRNA disease node pairs such as neighboring topology representations, however, may not be integrated and explicitly preserved in the above deep neural network models. In this work, we propose and develop a prediction model, named GTAN, to retrieve lncRNAs that are closely associated with a given disease. We first extract topological features and attribute matrices in a graphical representation of the given data by embedding the associations, similarities and interactions among lncRNAs, diseases and RNAs. We then construct GTAN to learn an attentional representation of lncRNA disease pairs including neighbor topology representation and integrated shallow and deep attributes representation. Finally, we determine top-ranked lncRNA candidates for the disease based on the scores of the output association prediction. The major contributions of our model include: we propose an embedding mechanism to integrate the topological information of a lncRNA, or disease, with its top-ranked similar neighbors, which is beneficial to learning the neighbor topology of node. Moreover, the embedding mechanism is formulated on the biological premise that a lncRNA, disease, and its most similar neighbors are usually associated with the same diseases, lncRNAs. Since different neighbors of lncRNA, disease, nodes contribute to association prediction differently, we establish a neighbors level attention mechanism, N-attention, to extract more informative neighbors of the nodes in the graph representation. Meanwhile, as there are interdependencies among multiple local topology representations of lncRNA, disease, nodes, we propose a topology level attention mechanism, T-attention, to enhance topology representation. N-attention and T-attention are used by encoding layers in GNN to reveal complex connections of lncRNA disease pairs from correlated lncRNA, disease and mRNA. We propose an attribute-level attention, A-attention, embedding mechanism to discriminate and integrate various contributions of different attributes, including pairwise lncRNA disease similarities and correlations. The mechanism is based on the premise that if a pair of lncRNA and disease has similarities, associations or interactions with more common lncRNAs, diseases and RNAs, then the lncRNA disease pair is more likely associated with each other. We introduce an encoding module based on multilayer CNN to capture and encode attribute representations of lncRNA disease pairs. The shallow detailed features and intricate representative features of pairwise nodes are learned by shallow and deep networks, respectively. The integration of shallow features can effectively make up for the missing details in the deep learning process. 2. Materials and methods. Given multi-source data related to lncRNAs and diseases, we design a GTAN model for predicting lncRNA disease associations based on GNN with attention mechanisms and CNN, Figure 1. We first introduce two embedding mechanisms for neighboring topologies and pairwise attributes to integrate the associations, similarities and interactions among lncRNAs, diseases and mRNAs. We then construct two branches in GTAN. Given lncRNA Li and disease DJ as an example, one branch captures the neighboring topology of Liege and the other branch is a multi-layer CNN module to encode the details and representative attributes of the Liege pair. The prediction scores from the two branches are combined by a weighted sum to score the corresponding association, which reflects the possibility of the lncRNA participating in disease processes. A dataset. The dataset is from 28, which was originally collected from lncRNA-disease database, 41, Starbasev 2.0 database, 42, Human microRNA disease database, HMDD, 43. lncRNA-disease provides the mappings between diseases and lncRNAs, Starbase F2.0 includes the information of the interactions between lncRNAs and mRNAs, and HMDD serves as an auxiliary tool for screening disease-related mRNAs. In total, 240 lncRNAs, 405 diseases and 495 mRNAs are analyzed, covering 2,687 lncRNA disease associations over 240 lncRNAs and 405 diseases, 1,002 interactions between 240 lncRNAs and 495 mRNAs, and 13,559 mRNA disease associations. Authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. Xuan et al. Graph Triple Attention Network for Disease-Related lncRNA Prediction 2841 Figure 1. Framework of the proposed GTAN model. Given lncRNA L1 and disease D2, a topology feature embedding and attribute embedding matrix of lncRNA and disease pairs are firstly performed. B. The first branch of GTAN learns the neighbor topology representation of lncRNA and disease nodes by N-attention and T-attention enhanced encoding layers. C. The second branch learns attribute representation of lncRNA disease pair with A-attention. D. Topology and attribute representation are finally integrated to obtain output association score of L1D2. Figure 2. Matrix representation construction from multi-source data related to lncRNAs in diseases. B. Multi-source data representation of lncRNAs in diseases. We first construct matrix representations of lncRNA and disease from multi-source data including lncRNAs, diseases and mRNAs as shown in Figure 2. The matrix representations include inter-lncRNA disease associations, inter-mRNA disease associations, inter-lncRNA-mRNA interactions, intra-disease similarities, and intra-lncRNA similarities. 1. Representation of lncRNA disease association and mRNA disease association. The associations of lncRNAs in diseases and mRNAs in diseases are represented by association matrix A equals lncRNA minus disease RND times ND, mRNA minus disease RNM times ND. 1. lncRNA minus disease records the associations between NL lncRNAs and ND diseases, where each row of lncRNA minus disease represents the associations between a lncRNA and all diseases. The ITH column of lncRNA minus disease contains the correlations between all lncRNAs and the ITH disease. lncRNA minus disease IJ is 1 if lncRNA Li and disease DJ are known to be associated, or 0, otherwise. Similarly, mRNA minus disease represents mRNA diseases associations between NM mRNAs and ND diseases, where mRNA minus disease IJ is 1 if mRNAs MJ and diseases DJ are related, or 0, otherwise. 2. Representation of lncRNA-mRNA interactions. Since most lncRNAs and mRNAs may interact and are involved in the same disease, 44-47, the interactions between NL lncRNAs and NM mRNAs are represented by the matrix lncRNA minus mRNA RNL times NM. lncRNA minus mRNA IJ equals 1 indicates authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. 2842 IEEE Journal of Biomedical and Health Informatics, Volume 26, Number 6, June 2022 that lncRNA Li and mRNA MJ interact, otherwise there is no known interaction relationship, lncRNA minus mRNA IJ equals 0. 3. Representation of disease similarities and lncRNA similarities. The similarity matrix of intra-disease and intra-lncRNA is defined as follows: S equals S disease RND times ND, SLNC RNA times NL. 2. Where the similarity matrix disease equals disease IJ, RND times ND represents the semantic similarities of diseases and ND is the number of diseases. We follow Wang et al. 48. To calculate the semantic similarity between two diseases through a directed acyclic graph of diseases. For disease DJ, the disease terms related to DJ constitute a directed acyclic graph. Values in disease age are between 0 and 1. A higher disease age value indicates a greater similarity between D and DJ. When calculating intralincRNA similarities, we exploit the similarity measure proposed by Chen et al. 18, which was based on the assumption that two lncRNAs would be more similar if more common diseases were associated with them. For instance, suppose that lncRNA Li is associated with a group of diseases D equals B1, D2, DIM, and lncRNA LJ is associated with DJ equals DJ1, DJ2, DJN, then the similarity between D and DJ is calculated, and the result is taken as the similarity between Li and LJ. See embedded representation extraction given the association, similarity and interaction matrices lncRNA-disease, mRNA-disease, lncRNA, disease and lncRNA-mRNA of lncRNAs, diseases and mRNAs. We introduce two embedding strategies to extract topology feature and pairwise attribute of lncRNA and disease from multi-source data, as shown in Figure 3. 1. Topology feature embedding. We propose a biological premise-driven embedding mechanism to extract the topological information of lncRNA and disease nodes from lncRNA and disease-related data, respectively. We assume that if two lncRNAs are more similar, they are more likely to contribute to similar diseases and interact with similar mRNAs. Likewise, similar diseases are more likely to be associated with similar lncRNAs and mRNAs. Based on the similarities between lncRNA Li and all the other lncRNAs, we calculate top NK neighbors that are most similar to Li. Similarly, we can get NK neighbors that closest to disease DJ. Let U be the topology vector of a lncRNA node and V the topology vector of a disease node. UI contains the topological relationships between Li and each of lncRNAs, diseases and mRNAs. Suppose NK equals 3 and SLI preserve 3 top-ranked neighbors LA, pound and LC of Li with topology vectors UA, UB and UC. The topology embedding SLI of Li is obtained as SLI equals UI, UA, UB, UC. 3. Figure 3. Illustration of topology feature embedding and attribute embedding matrix extraction process by L1 and D2 node, where UI is obtained by splicing the ITH-RAV matrix lncRNA, lncRNA-disease and lncRNA-mRNA, as UI equals lncRNA lncRNA-disease lncRNA-mRNA UIRNF. 4, where lncRNA denotes the ITH-RAV lncRNA, which records the similarities between Li and every lncRNA. lncRNA-disease is the ITH-RAV matrix lncRNA-disease containing the associations of Li with all diseases. lncRNA-mRNA contains the interactions between Li and each mRNA. NF equals NL plus ND plus NM, where NL, ND and NM are the numbers of lncRNAs, diseases and mRNAs, respectively. Represents the concatenation operation from beginning to end. Similarly, suppose that the top NK equals 3 neighbors of disease DJ are D, DF and DG. The neighbor topology embedding of DJ is SDJ, SDJ equals VJ, VE, VF, VG. 5, where VJ is the topology vector of DJ, which is defined as VJ equals lncRNA-disease *, Jace disease *, J mRNA-disease *, J. VJRNF. 6, where lncRNA-disease *, J, disease *, J and mRNA-disease *, J records the diverse connections between DJ and every lncRNA, disease and mRNA node. 2. Attribute embedding matrix of lncRNA disease pairs. If lncRNA Li and disease DJ have similarities or correlations with more common lncRNAs, diseases or mRNAs, it is more possible that Li and DJ are associated. Therefore, it is necessary to learn the attribute information of lncRNAs and diseases from the pairwise authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. Xuan et al. Graph Triple Attention Network for Disease-Related lncRNA Prediction 2843 Node Level. We express the similarity relationships between Li and every lncRNA by an attribute vector C1, the association between Li and all diseases by C2, and interactions between Li and every mRNA by C3. The relationships between disease DJ and every lncRNA, disease and mRNA are recorded in vector C4, C5 and C6, respectively. Attribute vectors CT, T equals 1, 2, 3, 4, 5, 6 are spliced to derive the attribute embedding matrix Z of Li and DJ as below Z equals C1, C2, C3, C4, C5, C6. Z are 2 times NL plus ND plus NM, 7, where 2 times NL plus ND plus NM is the dimension of ZD neighboring topology encoding module based on attention-enhanced graph neural networks. We propose an attention-enhanced GNN, ATTEN-GNN, to learn the neighbor topology representation shown in Figure 1b. Given the neighbor topology vector sets Li and SDJ of lncRNA Li and disease DJ, neighbor topology representation of Li node, DJ node, is obtained via its NK neighbors. Since the learning framework for lncRNAs is similar to that for diseases, we use lncRNA Li as an example to illustrate ATTEN-GNN. Given Li equals UI, UN, N equals A, B, C, since the topology vectors UI and UN are sparse, we firstly design a transformation matrix to project each node's vectors to a low-dimensional feature space. The projection process is given as follows: AT equals utlink, WLNCRNF times ing, 8, where AT is the projection of the original topology feature AT, which is a node in sets Li, and ing is the dimension of the low-dimensional feature space. 1. Neighbor-level self-attention mechanism, and attention, as not all neighbors of Li contribute equally to the learning of local topology representation of Li, we propose an attention to learn the attention weight of each neighbor as IN equals GTAN H. Node middle dot, UI vertical bar UNT. 9. Where UN denotes the low-dimensional topological vector after the projection of any neighbor node lane of Li, IN denotes the importance of a neighbor lane in the representation of Li. Node is the weight matrix. Vertical bar denotes the concatenation between the two vectors UI and UN, and G captures the context between multiple neighbors of Li. The attention-weighted neighbors are normalized by the softmax function, and the attention coefficient alpha n of each neighbor is obtained. Alpha n equals exp, IN, NKM equals 1 exp, AIM. 10. Finally, the weighted sum of the topological feature of the neighbors and its weight coefficient is transferred to the lncRNA Li, and the local topology representation of Li as P equals sigmoid N NK alpha and UN. 11. 2. Topology level attention, T attention. Since interdependencies exist between local topology representations of different levels, we establish T attention. In order to stabilize the learning process of N attention and capture rich neighboring topology information, we use multi-head attention, 49, and extend N attention to H head. Specifically, we stack HN attentions together. Thus, the local topology representation PH, to be learned by the HTHN attention, is taken as a specific response to the neighbor topology of Li from the HTH perspective. Given the HTH local topology representation PH of Li, we firstly calculate the correlations OM by performing vector multiplication followed by softmax between MTH local topological representation PM and PH as follows: OM equals exp PHPTM, HM equals 1 exp PHPTM, 12, where PTM is the transposed vector of PM. Next, we calculate the enhanced topology vector PH of Li as PH equals HM equals 1, plus PH. 13. Finally, we obtain neighbor topology representation T1 lnc of Li by splicing H topology representations from head to tail. T1 lnc equals H vertical bar H equals 1 sigmoid, pH 14, where vertical bar denotes the vector concatenate operation. The association prediction framework of the first branch is composed of multilayer A10 GNN. The output of the first A10 GNN layer, T1 lnc, is fed into the second layer GNN layer 2. GNN layer 2 performs similar learning process as GNN layer 1, which include low-dimensional feature learning, N-attention and T-attention, and generates node topology representation T2 lnc as output. Given T2 lnc as input, GNN layer 3 integrates H topology representations pH as TLNC equals sigmoid 1 HHH equals 1 pH. 15. The topology representations extracted by T-attention are first averaged and then processed by the sigmoid function to get the final neighbor topology representation TLNC of Li. The obtained neighbor topology representations of disease DJ are denoted by Ts. We also send SDJ to A10 GNN to obtain the neighbor topology representation of DJ as Ds. 3. Prediction score calculation in the first branch. Given TLNC and Ts learned from A10 GNN, neighborhood topology representations topo of a lncRNA-disease pair is obtained by concatenating TLNC and Ts. Then stopo is sent to a fully connected layer flat 1 and softmax layer to get itopo. Itopo equals softmax, flat 1 stopo plus b flat 1, 16, where itopo is associated probability distribution, and b flat 1 is bias vector. Y1 topo and Y0 topo denote the probabilities that Li are associated and unassociated with DJ, respectively. Authorized license to use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. 2844 IEEE Journal of Biomedical and Health Informatics, Volume 26, Number 6, June 2022 pairwise node attribute encoding based on CNN shallow and deep pairwise node attributes extraction as illustrated in Figure 1c. In this branch, we first feed the attribute matrix Z of lncRNA Li and disease DJ into a newly proposed attribute level attention module to obtain Z, followed by the first convolution module to obtain shallow level representation M1. To capture in-depth features, two convolution modules are used to obtain a deep feature map M2. 1. Attribute level attention, A attention. The attribute matrix Z contains 6 types of attributes, C1, C2, C3, C4, C5, C6. Since each attribute has distinct importance to association prediction, we establish an attention mechanism to obtain important weighted attributes. For attribute vector C where I, 1, 6, its attention score C is obtained as C equals hot phi, WATC plus BAT, 17, where hot and WA are weight matrices, BAT is a bias vector, and phi represents activation function 10H. The normalized attention weight beta ij of attribute value SIG is obtained as beta ij equals exp, shij, kexp, sic. 18. Finally, a attention enhanced attribute representation Re is obtained as Re equals cc, 19, where denotes elementwise multiplication operation. The feature matrix weighted by attention mechanism is represented by Z equals R1 R2 R3 R4 R5 R6. 22. Attention enhanced shallow and deep attribute integration. Firstly, let ZRL times H denote 0 padded Z where L and H are the numbers of rows and columns. Then Z is fed to the convolution layer and pooling layer to obtain shallow features M1 of Liege. Filters BONV1 RNC times NW times NH perform convolution operations on Z where NC is the number of filters, and NW and NH are the width and length of the filters. Then, ZK, I, J is the I, J, position covered by the KTH filter defined as ZK, I, J equals Z, I plus NH, J plus NW, ZK, I, JRNH times NW, 21, mConv1, Ki, J. The output from the KTH filter conv1 is obtained as mConv1, Ki, J, equals phi. Conv1, K, smile asterisk ZK, I, J plus bconv1, K, I, 1, L minus NW plus 1, J, 1, H minus NH plus 1, K, 1, and C, 22, where conv1 and conv1 are the weight matrix and bias vector respectively. The convolution operation performed by the filter on the feature map is represented by the symbol asterisk, and phi is a nonlinear activation function. A max pooling layer is used to extract a feature map M1 with details of liege node pairs. Then, M2 is obtained by 2 layers of convolution pooling operations to include more representative features. 3. Prediction score calculation in the second branch. The corresponding elements of the shallow feature map M1 and the deep feature map M2 are added and then flattened to obtain a vector Zatter. The association prediction score Yatter is acquired by passing Zatter through a fully connected layer and softmax layer as Yatter equals softmax, W flat 2 Zatter plus B flat 2, 23, where W flat 2 and B flat 2 are the weight matrix and bias vector of the fully connected layer, and Yatter is the associated probability distribution of the second branch. Final integration and optimization when learning neighbor topology representation in the first branch. The loss function is cross-entropy between the real label Z and predicted association probability I topo defined as loss topo equals minus train I equals 1 Cj equals 1 Zj log, I topo, Ja, 24, where train is a group of training samples, and predicted results are associated and unassociated, C equals 2. Cross-entropy lossature for the second branch is formulated as lossature equals minus train I equals 1 Cj equals 1 Zj log, Yatter, J. 25. We train the loss functions loss topo and lossature separately until each of them reaches a minimum. The final correlation prediction score Y is obtained as Y equals gamma times I topo plus 1 minus gamma times Yatter, 26, where gamma, 0, 1, is hyperparameter to adjust the contributions of the neighbor topology and pairwise attribute. 3. Experimental results and discussions. We perform systematic evaluations and comparisons to validate the performance of our model, including ablation experiments, evaluation of AUC, OPER, and top K recall rate over all 405 diseases and 10 well-characterized diseases, case studies of 3 common cancers, and enrichment analysis of candidate lncRNAs. Parameter settings for the neighbor topological encoding module, the number of A10 GNN encoding layers and the number of heads of multi-attention H are all set to 3. The dimensions of output feature space from the 3 encoding layers are 1100, 800 and 600. The attribute encoding module of lncRNA-disease pairs contains 3 convolution pooling layers, and the window size of filters in convolution and pooling operations was set as 2 times 2. The first convolution module has 16 filters, and those in the second and third modules have 32 and 16 filters, respectively. Our method is implemented in the PyTorch framework using one GPU card, NVIDIA GeForce GTX 2080 Ti. Authorized license to use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. Xuan et al., Graph Triple Attention Network for Disease-Related lncRNA Prediction 2845b Performance Evaluation Metrics. We perform 5-fold, 10-fold and 20-fold cross-validations to evaluate the performance of our new method and the 9 existing methods for predicting lncRNA-disease associations. Each of the 5-fold, 10-fold and 20-fold cross-validations is performed 50 times. We take the 5-fold cross-validation as an example to introduce the separation of training set and testing set. A total of 2,687 known associated pairs of lncRNA-disease are taken as positive samples and 94,513 unknown associations are used as negative examples. For each fold of the cross-validation, the training set consists of 4/5 positive samples and the same number of randomly selected negative examples. The remaining 1/5 positive samples and negative examples are used for testing. In each fold, the similarities between lncRNAs are recalculated using the involved positive samples. Evaluation metrics include receiver operating characteristic, ROC, curve, area under rock, AUC, 50, true positive rate, TPR, false positive rate, FPR, precision recall, PR, curve, 51, the area under PR, AUPR, and top K recall rates. TPR and FPR are defined as TPR equals TP-TP plus FN, FPR equals FP-TN plus FP, 27, where TP-TN denote the number of correctly predicted positive, negative, samples, FP-FN are the number of incorrectly predicted positive, negative, samples. Greater AUC values indicate better prediction performance. As there are imbalanced distributions between positive and negative samples, PR curve is also used to evaluate the performance. Precision and recall are calculated as precision equals TP-TP plus FP, recall equals TP-TP plus FN, 28, where precision indicates the proportion of real positive samples that are predicted as positive. Recall reflects the rate of correctly identified positive samples among all positive samples. Top K recall rates are calculated since biologists often take the top-ranked lncRNA-disease pairs for further verification. In each fold of cross-validation, for a disease DJ, we rank the candidate lncRNAs in a descending order of their association scores. Let LM-DJ node pair be a positive sample, and lane-DJ a negative sample. Given a threshold theta, if the predicted score of LM-DJ is greater than theta, it is deemed as a correctly predicted positive sample. If the predicted score of lane-DJ is smaller than theta, it is taken as a correctly determined negative sample. By changing the threshold theta, we calculate the TPR, FPR and recall rate to obtain the ROC curve and PR curve of DJ, as well as AUC and OPR. In the 5-fold cross-validation experiment, 5 AUCs and OPRs of DJ can be obtained separately. We then calculate the average AUC, AUPR, for every disease. The average AUC and OPR on all 405 diseases are used to quantify the overall prediction performance. See ablation experiments. We perform ablation experiments to validate the contributions of major innovations including neighbor-level attention table I ablation study results on our method, N attention, topology level attention, T attention, attribute-level attention, A attention, and shallow attribute information to the prediction of lncRNA-disease associations. As shown in Table I, the final model GTAN, with triple attention mechanisms and integrating shallow attributes achieved the best performance. Without N attention, the performance decreased by 1.9% in terms of AUC and 4.3% in terms of OPR. Without T attention, or A attention, the AUC and OPR dropped down by 0.7% and 1.7%, or 2.1% and 8.2%, when compared with GTAN. The ablation study demonstrated the effectiveness and contributions of the triple attentions. We also conducted ablation experiments to verify the contribution of the shallow attributes to the learning of node attribute representation. Compared with the model without shallow detail attributes, our final model GTAN increased the performance by 1.7% and 5.4% in terms of AUC and OPR, which indicated that shallow attributes effectively improve the prediction performance. Comparison with other methods. Our model GTAN was compared with some of the state-of-the-art methods for lncRNA-disease association prediction, including VADLP, 38, KNDA, 34, SIMPLA, 26, Ping's method, 22, MFLA, 28, LAP, 20, GAN, 39, GAMLA, 40, and LRLSLA, 17. GTAN and all the 9 methods in comparison are trained and validated using the same training and testing datasets in the cross-validation process. The ROC and PR curves of all the methods in comparison over 405 diseases are shown in Figure 4. As shown, our model achieved the highest average AUC of 0.983, outperforming VADL by 3.8%, TINDA by 5.0%, SIMCLA by 23.9%, Ping's method by 10.8%, MFLDA by 35.8%, LDAP by 12.2%, GAN by 4.3%, GANLA by 2.0%, and LRLSLA by 18.6%. In terms of PR, GTAN achieved the best average OPR of 0.454, which was 8.9%, 17.4%, 39.2%, 30.1%, 41.1%, 28.4%, 15.4%, 4.9%, and 25.4% higher than VADL, TINDA, SIMCLDA, Ping's method, MFLDAP, LDAP, GAN, GANLA, and LRLSLA, respectively. We also list the AUCs and OPRs of all the methods on 10 well-characterized diseases that are associated with at least 15 lncRNAs in supplementary tables. SD1 and SD2.1 GTAN achieved the best AUC over 9 of the 10 diseases and best OPR over 8 of the 10 diseases. We explained the improved results by neighboring topology and attribute representation of lncRNA disease pairs. All supplemental files are available at https://github.com/pingxuanliju/GTAN. Authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. 2846 IEEE Journal of Biomedical and Health Informatics, Volume 26, Number 6, June 2022. Figure 4. ROC and PR curves of lncRNA disease association prediction by different methods. Table 2. A pairwise comparison by a paired Wilcoxon test on the prediction results. The contributions of neighboring topology can be demonstrated by comparing our model and the other methods compared. The effectiveness of attribute representation was shown by the experimental results that our model and the third best VADLP achieved better performance than the other 7 methods. The primary reason is that deep CNN can capture diverse and complex correlations between lncRNAs and diseases. Besides, the AUC and OPR of Ping's method and LDAP were similar, which are both better than MFLDA. The main reason was that the latter did not take lncRNA similarities and disease similarities into consideration. Even though GAN and GANLA utilized deep learning methods, they ignored detailed features and representative features of node pairs so that they had inferior performance than GTAN. We also performed a paired Wilcoxon test to validate whether the improvement by GTAN was statistically significant. For each of 405 diseases, we first calculate its AUC and OPR under each fold cross-validation. The AUCs, OPRs, of 5 folds of each disease are averaged as its final AUC, AUPR. For each prediction method, we obtain 405 AUCs, OPRs, of 405 diseases. When comparing 2 methods, we perform paired Wilcoxon tests by using 405 paired AUCs, OPRs. The results are given in Table 2 as shown. AUCs and OPRs have a p-value less than 0.05, which higher than those of other methods. The top K recall rates are given in Figure 5. A higher recall rate means that more lncRNAs related to the disease are Figure 5. The average recalls of prediction methods at different top K cutoffs. Correctly identified. As shown, when K increases from 30 to 120 with an increment of 30, the average recall rates of GTAN are 90.7%, 94.1%, 95.0% and 98.2%, which is consistently better than all the other methods. VADL ranks 83.7%, 90.8%, 92.1%, and 94.0% in top 30, 60, 90, and 120, respectively. The third best method is CNDA with recall rates of 70.2%, 89.5%, 90.4% and 91.8%. When K is 30, Ping's method's recall rate is higher than that of LAP. However, when K is 60, Ping's method becomes inferior to LAP. Simpla has recall rates of 46.3%, authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. Xuan et al. Draft Triple Attention Network for Disease-Related lncRNA Prediction 2847 Table 3 Top 15 Lung Cancer-Related lncRNA Candidates 60.8%, 75.0% and 82.3%, which are slightly higher than those of mFLDA. GANLA achieved higher recall rates than GAN. The former ranks 87.4%, 92.4%, 94.2% and 98.0% in top 30, 60, 90 and 120, respectively, and the latter ranks 82.3%, 89.7%, 91.6% and 94.7%. LRL-SLDA is not as good as GAN and GANLA, and the corresponding recall rates are 49.6%, 63.8%, 75.2% and 84.9%. We conducted 10-fold cross-validation and 20-fold cross-validations to further evaluate the performances of the prediction methods when the training set contains various numbers of samples. The 10-fold and 20-fold cross-validations were performed for 50 times respectively, and the corresponding results are listed in the supplementary tables ST3 and ST4. We also showed the performances of GTAN in supplementary figure F1. The experimental results indicate that our method achieves better performance than the other methods. In terms of 10-fold cross-validation, GTAN achieved 3.9%, 5.2%, 23.5%, 10.9%, 35.6%, 12.1%, 4.4%, 2.0% and 18.5% higher EUCs than VADLP, KNDA, SIMCLDA, Ping's method, mFLDA, LAP, GAN, GANLA and LRL-SLDA, respectively. The APRs of GTAN increased by 9.4%, 17.3%, 39.3%, 30.4%, 41.3%, 28.7%, 15.2%, 5.1%, and 25.4% compared with the existing methods, respectively. For the 20-fold cross-validation, GTAN achieved 0.990 of AUC and 0.587 of APR. It is 3.8%, 5.4%, 23.5%, 10.9%, 35.7%, 12.1%, 4.4%, 2.0% and 18.7% higher than the other methods, respectively. GTAN's APR increased by 8.8%, 17.1%, 39.1%, 30.1%, 41.5%, 28.6%, 15.1%, 5.0% and 25.6%, respectively. For most of the prediction methods, the performance of the 20-fold cross-validation is slightly better than that of the 10-fold. One possible reason is the former use more samples for model training than the latter. The experimental results show that when the number of training samples increases, GTAN consistently achieved better prediction performance than the other methods. Our GTAN method outperformed the other methods compared for the following primary reasons. First, GTAN encodes and learns the enhanced neighbor topology representations of lncRNA nodes and disease nodes by graph neural network with neighbor-level attention and topology-level attention. Second, the topology-level attention mechanism captures the interdependence among multiple topological representations from multi-head learning. Finally, GTAN's multi-layer convolutional neural network module deeply integrates the detailed features and the representative features of lncRNA disease node pairs. In addition, The attribute-level attention mechanism is able to distinguish the differential contributions of different attributes for the association prediction. Case studies, lung cancer, prostate cancer and colon cancer. To further demonstrate GTAN's ability to detect lncRNA-disease associations, we performed case studies on 3 common diseases, i.e., lung, prostate, and colon cancer. The candidate lncRNAs are ranked in descending order of prediction scores for each of these cancers. The top 15 candidate lncRNAs for the 3 diseases are listed in tables 3, 4, and V. The top 15 candidates related to lung cancer are listed in table 3. LNC2 cancer includes the relationships between lncRNAs and human cancers that have been verified by biological experiments, 52. Among the top 15 lncRNA candidates associated with lung cancer, 9 candidates are confirmed by LNC2 cancer, which means that these lncRNAs are significantly upregulated or downregulated in lung cancer. lncRNA-disease provides experimentally supported associations of lncRNAs and disease, which are mined from the published papers, 41. Besides, lncRNA-disease includes 10 lncRNAs of the top-ranked candidates. The associations between lncRNA candidates, Hulk and MIR155Hg, and lung cancer are supported by the results in 53 and 54. Among them, MIR155Hg affects the proliferation and migration of human lung cancer cells through the production of MIR155-5P. As shown in Table 4, 11 out of 15 lncRNA candidates associated with prostate cancer from our model are supported by both LNC2 cancer and lncRNA-disease database. It indicated that these candidates are indeed associated with prostate cancer. As shown in Table V, 13 candidates of colon cancer appear in LNC2 cancer, and lncRNA-disease verifies 3 predicted lncRNA candidates. Among all the predictions, only 1 lncRNA candidate is not confirmed by observational evidence. Investigation of novel disease-related lncRNAs prediction. Finally, we applied the trained GTAN model to predict new lncRNA disease associations. The top 50 potential lncRNA candidates selected by our model are given in supplementary table SD5, which can be used to assist biologists for further experimental validation. Authorized licensed use limited to University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. 2848 IEEE Journal of Biomedical and Health Informatics, Volume 26, Number 6, June 2022. Table 4 Top 15 Prostate Cancer-Related lncRNA Candidates. Table V Top 15 Colon Cancer-Related lncRNA Candidates. 4. Conclusion. We proposed a deep learning model to discover candidate lncRNA disease associations. GTAN integrates the similarities of intra-lncRNAs and intra-diseases and the associations and interactions of inter-nodes, including lncRNAs, diseases and mRNAs. Neighbour and topology enhanced GNN and multilayer CNN learn and encode the neighbour topology representation and attribute representation from pairwise lncRNA disease nodes. Triple-level attention mechanisms adaptively extract and integrate informative neighbors, topology and attributes. Experimental results demonstrated that GTAN improved the overall prediction results and identified top-ranked lncRNA candidates for diseases. Our model has the potential to serve as a tool for discovery of real lncRNA disease associations through wet lab experiments. References: 1. M. Stellar, Non-Coding RNAs in Human Disease, Nat. Reverend Jennett, Volume 12, Number 12, pages 861 to 874, 2011. 2. T. R. Mercer, M. E. Diener, and J. S. Maddock, Long Non-Coding RNAs, Insights into Functions, Nat. Reverend Jennett, Volume 10, Number 3, pages 155 to 159, 2009. 3. M. Kitagawa, K. Kitagawa, Y. Kotake, H. Niida, and T. Ohata, Cell Cycle Regulation by Long Non-Coding RNAs, Cellular Mole. Life Sci. Volume 70, Number 24, pages 4785 to 4794, 2013. 4. Xley et al., A Comprehensive Survey on Computational Methods of Non-Coding RNA and Disease Association Prediction, Brief. Bioinf. Volume 22, Number 4, pages 1 to 31, 2021. 5. E. Anastasiadou, L. S. Jacob, and F. J. Slack, Non-Coding RNA Networks in Cancer, Nat. Reverend Cancer, Volume 18, Number 1, pages 5 to 18, 2018. 6. J. Li, Z. Xuan, and C. Liu, Long Non-Coding RNAs and Complex Human Diseases, int. J. Mol. Sci. Volume 14, Number 9, pages 18790 to 18808, 2013. 7. X. Chen, C. C. Yan, X. Zhang, Ans. H. U. Long Non-Coding RNAs and Complex Diseases, From Experimental Results to Computational Models, Brief. Bioinf. Volume 18, Number 4, pages 558 to 576, 2017. 8. B. Signal, B. S. Gloss, and M. E. Diener, Computational Approaches for Functional Prediction and Characterization of Long Non-Coding RNAs, Trends Genet, Volume 32, Number 10, pages 620 to 637, 2016. 9. M. X. Liu, X. Chen, G. Chen, Q. H. Kui, and G. Y. Yan, A Computational Framework to Infer Human Disease-Associated Long Non-Coding RNAs, PLOS 1, Volume 9, Number 1, 2014, Art. No. E84408. 10. G. Chen et al., lncRNA-disease, A Database for Long Non-Coding RNA-Associated Diseases, Nucleic Acids Res, Volume 41, No. D1, pp. D983-D986, 2012. 11. X. Chen, Y. A. Huang, X. S. Wang, Z. H. U. and K. C. Chan, FEMLNCSIM, Fuzzy Measure-Based lncRNA Functional Similarity Calculation Model, OncoTarget, Volume 7, Number 29, 2016, Art. Number 45948. 12. M. B. Clark et al., Genome-Wide Analysis of Long Non-Coding RNA Stability, Genome Res, Volume 22, Number 5, pages 885 to 898, 2012. 13. J. Li et al., A Bioinformatics Method for Predicting Long Non-Coding RNAs Associated with Vascular Disease, Sci. China Life Sci, Volume 57, Number 8, pages 852 to 857, 2014. 14. X. C. Lin et al., Integrated Analysis of Long Non-Coding RNAs and RNA Expression Profiles Reveals the Potential Role of lncRNAs in Gastric Cancer Pathogenesis, int. J. Oncol, Volume 45, Number 2, pages 619 to 628, 2014. 15. X. Chen, Katz Measure for the lncRNA-Disease Association Prediction, Sci. Rep. Volume 5, 2015, Art. Number 16840. 16. A. K. Biswas, B. Zhang, X. Wu, and J. X. Gao, A Multi-Label Classification Framework to Predict Disease Associations of Long Non-Coding RNAs, lncRNAs, in Proc. 3rd int. Conference Common, Signal Process, SIST, Springer, 2015, pages 821 to 830. 17. X. Chen and G. Y. Yan, Novel Human lncRNA Disease Association Inference Based on lncRNA Expression Profiles, Bioinformatics, Volume 29, Number 20, pages 2617 to 2624, 2013. 18. X. Chen, C. C. Yan, C. Luo, W. G. Weixiong, and Q. Dai, constructing lncRNA functional similarity network based on lncRNA disease associations and disease semantic similarity, Sci. Rep. Volume 5, 2015, Art. Number 11338. 19, Y. A. Huang, X. Chen, Z. H. U. D. S. Huang, and K.C. Chan, Ilngsim, Improved lncRNA Functional Similarity Calculation Model, OncoTarget, Volume 7, Number 18, 2016, Art. Number 25902. 20, W. Lan et al., VAP, A Web Server for lncRNA Disease Association Prediction, Bioinformatics, Volume 33, Number 3, pages 458 to 460, 2017. 21, J. Zhao et al., Construction of a Specific SVM Classifier and Identification of Molecular Markers for Lung Adenocarcinoma based on lncRNA mRNA Network, OncoTargets there, Volume 11, pages 3129 to 3140, 2018. Authorized Licensed Use Limited to, University of Illinois at Chicago Library. Downloaded on December 6, 2022 at 10:50 and 44 seconds UTC from IEEE Explorer. Restrictions apply. Xuan et al., Graph Triple Attention Network for Disease-Related lncRNA Prediction 2849, 22, P. Ping, L. Wang, L. Huang, S. Yi, M. F. B. Iqbal, and T. Pei. A novel method for lncRNA disease association prediction based on an lncRNA disease association network, IEEE/ACM Trans. Compute. Bioinf. Volume 16, Number 2, pages 688 to 693, March/April 2019. 23, J. U. Z. Xuan, X. Feng, Q. Zhou, and L. Wang. A novel collaborative filtering model for lncRNA disease association prediction based on the naive Bayesian classifier, BMC Bioinf, Volume 20, Number 1, pages 396 to 408, 2019. 24, Y. Li, J. Li, and NBN, NINF LDA. Prediction of lncRNA disease associations by dual network integrated logistic matrix factorization and Bayesian optimization, Genes, Volume 10, Number 8, pages 608 to 621, 2019. 25, X. Zhang, H. Ding, H. Mamitsuka, and S. Ju, collaborative matrix factorization with multiple similarities for predicting drug-target interactions, in Proc. 19th Akmzik Dint. Conference.nol. Discoff.data mining, 2013, pages 1025 to 1033. 26, C. Lu et al., Prediction of lncRNA disease associations based on inductive matrix completion, Bioinformatics, Volume 34, Number 19, pages 3357 to 3364, 2018. 27, Z. Xuan, J. Li, J. U, X. Feng, B. Zhao, and L. Wang, a probabilistic matrix factorization method for identifying lncRNA disease associations, Genes, Volume 10, Number 2, pages 126 to 145, 2019. 28, G. Fu, J. Wang, C. Domeniconi, and G. U. Matrix factorization-based data fusion for the prediction of lncRNA disease associations, Bioinformatics, Volume 34, Number 9, pages 1529 to 1537, 2018. 29, X. N. Fan, S. W. Zhang, S. Y. Zhang, K. Ju, and S. Lu. Prediction of lncRNA disease associations by integrating diverse heterogeneous information sources with RWR algorithm and positive pointwise mutual information, BMC Bioinf, Volume 20, Number 1, pages 1 to 12, 2019. 30. Q Yao et al. Global Prioritizing Disease Candidate lncRNAs via a Multi-Level Composite Network, Sci. Rep. Volume 7, Number 1, pages 1 to 13, 2017. 31. C. Gu et al. Global Network Random Walk for Predicting Potential Human lncRNA Disease Associations, Sci. Rep. Volume 7, Number 1, pages 1 to 11, 2017. 32. X Chen, Z. H. U. G. Y. Yan, and D. W. Gong, ERDA, Improved Random Walk with Restart for lncRNA Disease Association Prediction, OncoTarget, Volume 7, Number 36, 2016, Art. Number 57919. 33. J. Li, X. Li, X Feng, B Wang, B Zhao, and L Wang, a novel target convergence set-based random walk with restart for prediction of potential lncRNA-disease associations, BMC Bioinf, Volume 20, Number 1, pages 1 to 13, 2019. 34. P Xuan, Y Cao, T Zhang, R Kong, and Z Zhang, Dual Convolutional Neural Networks with Attention Mechanisms-Based Method for Predicting Disease-Related lncRNA Genes, Front. Genet, Volume 10, pages 416 to 426, 2019. 35. H. Wei, Q. Liao, and B. Lu, lncRNAs FB, identify lncRNA disease associations by fusing biological feature blocks through deep neural network, IEEE/ACM trans. Compute. Bio. Bioinf, Volume 18, Number 5, pages 1946 to 1957, September/October 2021. 36. P Xuan, H Kui, T Shen, N Sheng, and T Zhang, Hetero-Dual Net, a dual convolutional neural network with heterogeneous layers for drug disease association prediction via Cho's 5-step rule, Front. Pharmacal, Volume 10, pages 1301 to 1312, 2019. 37. P Xuan, S Pan, T Zhang, Y Lu, and H Sun, Graph Convolutional Network and Convolutional Neural Network-Based Method for Predicting lncRNA Disease Associations, Cells, Volume 8, Number 9, pages 1012 to 1027, 2019. 38. N Sheng, H Kui, T Zhang, and P Xuan, Attentional Multilevel Representation Encoding based on Convolutional Invariance Autoencoders for lncRNA Disease Association Prediction, Brief. Bioinf, 2021, Art. No. BO 67. 39. J Zhang, Z Zhang, X. Hu, and Yisong, a novel graph attention adversarial network for predicting disease-related associations, methods, Volume 179, pages 81 to 88, 2020. 40. W Lan, X. Wu, Q Chen, W Peng, J Wang, and Y.T. Chen, Dan Le, Graph Attention Network for lncRNA Disease Associations Prediction, Neurocomputing, Volume 496, pages 384 to 393, 2021. 41. Z Bao, Z Yang, Z Huang, Y Zhou, Q Kui, and D Dong, lncRNA Disease 2.0, an updated database of long non-coding RNA-associated diseases, nucleic acids REs, Volume 47, No. D1, pp. D1034 D1037, 2019. 42. J. H. Li, S. Lu, H. Zhou, L. H. Chu, and J. H. Yang, Starbase v2.0, Decoding miRNA-siRNA, miRNA-NCRNA and protein RNA interaction networks from large-scale ClipSec data, Nucleic Acids REs, Volume 42, No. D1, pp. D92 D97, 2014. 43. Z Huang et al., HMDD v3.0, a database for experimentally supported human microRNA disease associations, Nucleic Acids Res, Volume 47, No. D1, pp. D1013 D1017, 2019. 44, X. Chen, Predicting lncRNA Disease Associations and Constructing lncRNA Functional Similarity Network Based on the Information of mRNA, Sci. Rep. Volume 5, Number 1, pages 1 to 11, 2015. 45, Jiang, X. Liu, and L. Yuan, lncRNA, A Link Between RNA and Cancer, Biochimica et Biophysica Acta, BBA Gene Regulatory Mechanisms, Volume 1839, Number 11, pages 1097 to 1109, 2014. 46, M. D. Paraskevopoulos and A. G. Hatzagorgiou, Analyzing RNA Interactions, in Long Non-Coding RNAs. Berlin, Germany, Springer, 2016, pages 271 to 286. 47, N. Li, M. Panusami, M. P. Li, K. Wang, and P. F. Li, The Role of MicroRNA and lncRNA microRNA Interactions in Regulating Ischemic Heart Disease, J. Cardiovasc. Pharmacal Therapeutics, Volume 22, Number 2, pages 105 to 111, 2017. 48, D. Wang, J. Wang, M. Liu, F. Song, and Q. Kui, Inferring the Human MicroRNA Functional Similarity and Functional Network Based on MicroRNA-Associated Diseases, Bioinformatics, Volume 26, Number 13, pages 1644 to 1650, 2010. 49, P. Velikovic, G. Q. Kuril, A. Casanova, A. Romero, P. Leo, and Y. Bengio, Graph Attention Networks, in Int. Conference Learn. Representations, Vancouver, Canada, pages 1 to 12, 2018. 50, K. Haji and Tilaki, Receiver Operating Characteristic, ROC, Curve Analysis for Medical Diagnostic Test Evaluation, Caspian J. Intern. Med, Volume 4, Number 2, pages 627 to 635, 2013. 51, T. Saito and M. Reemsmeyer, The Precision Recall Plot is more informative than the ROC plot when evaluating binary classifiers on imbalanced datasets, PLOS 1, Volume 10, Number 3, 2015, Art. No. E01184332. 52, S. Ning et al., LNC2 Cancer, a manually curated database of experimentally supported lncRNAs associated with various human cancers, Nucleic Acids Res, Volume 44, No. D1, pp. D980-D985, 2016. 53, J. Zhang, S. Liu, J. F. Ju, and K. P. Yang. Upregulation of lncRNA Hulk predicts a poor prognosis and promotes growth and metastasis in non-small cell lung cancer, int. J. Klin. Exp. Pathol, Volume 9, Number 12, pages 12415 to 12422, 2016. 54, S. Balin and Y. Nengshu, Long Non-Coding RNA Mir 155 HE promotes proliferation, migration and invasion of A549 human lung cancer cells, J. Chongqing Med. University, Volume 42, Number 1, pages 21 to 26,